So now I'd like to introduce our first year GGI graduate student, Pamela Watson, to give the introduction.
Okay, awesome, thank you. Hello everyone. Just kind of a quick introduction. I don't want to eat into Dr. Dapper's time, but this year, the first and second year graduate students had the opportunity to nominate a speaker for the GGI Spring Seminar Series. We each voted and chose our speakers based upon our own interests, and Dr. Dapper was my nominee. Because of the current health situation, some speakers weren't available, so we are truly lucky to have her speaking virtually with us today. Dr. Dapper joins us today from Mississippi State University, where she is currently an assistant professor. Her lab studies reproductive diversity across many different species, from birds to nematodes to humans. She obtained her PhD from Indiana University and then went on to complete her postdoctoral training at the University of Wisconsin. Across her career, she has received training and research grants from the NSF and the NIH. Her work has been published in Science Advancements and Evolution, and she has given over 25 seminar talks. Once again, we are truly lucky to have her speak with us today, and if we could all hold the questions till the end, that would be appreciated. Thanks. Go ahead.
Oh, thank you very much, Pamela, for that introduction. That was very nice. And I want to say that I'm really deeply honored to be invited by the grad students. And I wish that I was able to be there and meet with you guys in person, because I'd love to hear about all the cool stuff and research that you're doing. And hopefully when we get through all of this, I'll be able to come up and visit and see more what you guys are doing there. So today I am going to talk about the evolution of meiotic recombination in mammals mostly, but also more generally in vertebrates, and how my lab group is taking a pathway perspective to generate new insights into how this really fundamentally important trait evolves. Oop. All right. There we go. So as an evolutionary biologist, what I really ultimately want to understand is how great diversity of life on Earth was generated. And so using this really beautiful picture of diversity in beetles to illustrate all the differences that we see among species, but we can talk about it not just in terms of morphological diversity, but in terms of genes and genetic diversity and physiological diversity and cellular processes as well. And to understand how these differences arise between species, ultimately we need to understand patterns of variation within species and how those patterns of variation within species arise, and the patterns that you see between different populations really is crucially important to understanding how differences between species arise. And so broadly, as Pamela mentioned, my lab is interested in how reproductive processes shape these patterns of variation, and specifically genetic variation within species. And we look at reproductive processes at lots of different levels of biological organization. So we're interested in, I'm gonna see if I can turn on my laser pointer here. Yeah. We're interested in behavioral variation, so how variation in mating system and who mates with who can shape patterns of variation within populations. We're also interested in looking at interactions between cells. So inside of the female reproductive tract, how are sperm interacting with other sperm and competing to fertilizing the eggs, and how do sperm and eggs themselves interact, and how that shapes how patterns of variation and what genetic variation is passed on to the next generation. And we're also interested in these processes that are occurring within these reproductive cells, within sperm and eggs, and how cellular processes like meiotic recombination, which I'm going to focus on today, determines what genetic variation is ultimately in those gametes that then fertilize in the context of cells and then are introduced to each other through behavioral interactions. So why do I think studying meiotic recombination is so important? So I'm going to take a step back and think about go back to cell biology, introductory cell classes. So what is meiosis? Meiosis is a special form of cell division that happens in diploid organisms, or organisms with two copies of homologous DNA, 2 copies of each chromosome that are maternally and paternally inherited. How did those diploid cells then divide to produce haploid gametes? And so each individual has to go through a process where they take specific cells in their body, and they go through this process of meiosis, which creates haploid gametes, and those haploid gametes are then transmitted to produce offspring. So within meiosis, in this very early stage of meiosis, you get the process of meiotic recombination, which is the physical exchange of genetic material between homologous chromosomes. And I'm gonna see if I can play this video here to sort of emphasize how beautiful and dynamic meiosis is. So there you go. So what's going on here, this is a spermatocyte cell in a crane fly, and you see along this center region are all of the chromosomes in this cell, and they're all lining up, and then they're gonna get pulled into different poles of this cell, which is then, if it kept going, divide and then go through another round of cell division. And as you saw in the beginning of the video, all of those pairs of homologous chromosomes were really nicely aligned in the center of that cell, which allowed the cell to then use the spindles to pull them apart. And meiotic recombination is really important for properly aligning those chromosomes, because those chromosomes have to figure out which is their matching partner, which is their homologous chromosome, and then line up in a really organized manner in order for them to be accurately pulled apart. And meiotic recombination really regulates and drives that homology search and matching between chromosomes. And so if you don't get the right amount of recombination, there's a lot of really large effects on fertility, and it can, in humans, most cases of infertility, or a very large proportion of cases of infertility, are due to errors in meiotic recombination. And it also can lead to improper numbers of chromosomes in cells, which is a cause of things like Down syndrome. So if you don't have the right amount of recombination, there are really important downstream effects on reproduction and development. But recombination is also a really important genomic and evolutionary parameter as well. So in addition to playing this key role in getting the right chromosomes and the right gametes, it also allows the generation of genetic diversity by the exchange of genetic material between chromosomes. And so you can get combinations of alleles on these gamete chromosomes that weren't present in the parental chromosomes. And that shapes patterns of genetic variation throughout the genome and determines the efficacy of selection. So it's really a fundamental genomic and evolutionary parameter. And it shapes patterns of variation within genomes because recombination rate isn't constant. It isn't constant at pretty much any biological scale that you look at. So if you look at the whole genome, down here, a little picture of the whole, or representation of a series of chromosomes. There's variation between individuals and between species and between populations, and the number of recombination events that happen each meiosis. But if you look in and zoom in to parts of chromosomes, you'll see there are hot spots and cold spots. Some of them are relatively large, but in a lot of species, you even get these really fine scale, KB type scale differences in recombination, which we call recombination hotspots, which are really prevalent in a lot of mammals. And these differences within the genome in the rates of recombination shape things like GC bias, what is the nucleotide composition, differences in codons usage, and transposable elements, and all sorts of things. So it has really important effects on the genomic landscape. But it also varies between species. And so it varies on all of these different levels, but this is a survey, a representation of a survey of differences in recombination rate among a really broad group of species. So you see over here we have conifers. Conifers have a really low recombination rate, whereas over here we see really high recombination rates in fungi. And we also see tremendous variation within different groups of organisms. So you can see fish here have tons of variation, as do insects. And then some of that pattern comes from differential sampling. So some of these, we don't have a lot of estimates of recombination rates for frogs and birds necessarily. But the real point of this slide is to show you that there's lots of variation between species and recombination rate. And despite recombination rate having such important effects at the cellular level, in terms of accurate chromosomal segregation, or at these evolutionary and genomic levels as well, we really don't have a good understanding of how evolution shapes these patterns of variation and recombination. And we don't even really know if this variation that we're seeing in recombination rate is adaptive, or if it increases the fitness of organisms. And so it's really exciting to be able to study a trait where it's so important, but we really don't know what's going on, because that means there's tons of cool things to learn. And so what I'm going to talk about today is one direction my lab is taking to try to generate new insight into the evolutionary forces that are shaping diversity and recombination rate. And so most generally, we can ask, what are the causes and consequences of variation in recombination rate? And specifically, we want to know, are there molecular changes that we can identify in genes that underlie this variation in recombination rate? And because these genes don't operate in a vacuum, they operate in a cellular context, we want to try to put them into the context of the pathway and ask if, by not only looking at molecular changes in genes, but understanding how those genes impact recombination and which genes those genes interact with, can we use this perspective to generate novel insight into how this trait evolves? So I want to start by talking about how recombination occurs, and what is this pathway that we want to put our results in the context of. So I'm going to start by just orienting you here. At the very beginning of meiosis, DNA duplicates. So we have two homologous sets of homologous chromosomes. We'll just say this one is paternally inherited and this one is maternally inherited. And it's duplicated, so there's two copies, double-stranded DNA copies of each homologous chromosomes, which we'll call sister chromatids. And the first thing that happens in recombination, which is really crazy in my opinion, is that the DNA comes in and it creates double-strand breaks throughout the genome. And this is just going to focus on a small portion of the genome, but they do this in humans and mice. It's 200 to 300 different times. And it just boggles my mind still that the beginning of meiotic recombination consists of damage. You really don't want to damage your DNA, because that can create all sorts of issues, can induce mutations, but that's exactly what's required for meiotic recombination to start. And then those double-strand breaks have to be identified. And then there's some other genes and proteins that come in and resect and expose some single-stranded DNA at each one of those cuts. And then there's a process of homology search and strand invasion. So when we make these slides and these figures, it just so happens that we are perfectly matching up the homology between the two chromosomes. But in the cell, if you think back to that video we saw, it's a really dynamic environment, and it's actually much more of a complex problem for this homologous chromosome to figure out which one in that sort of cellular milieu does it match up with. And so these exposed single-stranded DNA have a series of proteins that help them find where they match, where their sequence matches in their homologous pair. And then the process where they sort of insert themselves into the homologous chromosome is called strand invasion. So after that homology search and the chromosomes correctly pair up, there's stabilization of that pairing. And so there's this whole suite of proteins that come in and form what we call a synaptonemial complex. And that is a complex of proteins that links and stabilizes that pairing of those two chromosomes. And it importantly provides a substrate on which these recombination events happen. And so the next thing that happens is there's a crossover versus non-crossover decision. So I mentioned there's 200 to 300 double-strand breaks in the cell, but there are not 200 to 300 crossovers or recombination events. In mice and humans, it's closer to 20 or 30. So there's a whole suite of genes that come in and they designate which of those double-strand breaks is actually going to be resolved as a crossover, like you see here, or a non-crossover where it gets repaired and there's tiny bit of transfer of information between the two homologous chromosomes, but it's generally repaired without a large exchange of genetic material. And then those breaks in the DNA, those crossovers are resolved. And so here we see this tiny track of gene conversion that happens due to non-crossovers. And then we see here this exchange, large-scale exchange of genetic material in this instance where it resolved as a crossover. So this is just to highlight how the process of recombination funnels down these originally 200 to 300 double-strand breaks into 20 to 30 crossovers. And a lot of the action happens later in the pathway after the Synaptonemial Complex, or this stabilization of homologous pairing, has occurred, when those enzymes and proteins are recruited to determine which of those original 200 to 300 double-strand breaks get resolved as crossovers. And so what we wanted to ask is, what is the rate of evolution of recombination genes across the mammalian phylogeny? If we're able to tell what genes are involved in each of these steps, which ones are showing high levels of divergence in their sequence, and can we then learn something about how the phenotype that they regulate has evolved and diverged as well? And specifically, we wanted to know whether we observe higher rates of divergence in steps that we think are likely to determine recombination rate. So there's two hypotheses about where in this pathway you could get variation in the resulting phenotype. The first hypothesis is that you could get variation all the way up at the start of the pathway in terms of the number of double-strand breaks that form. So maybe it's the case that if you have more double-strand breaks, those differences sort of cascade through the entire pathway and you end up with more crossovers if you have more double-strand breaks. Or alternatively, maybe it's this last step, later steps of the pathway, where those double-strand breaks are determined to be either crossovers or non-crossovers, that really is most important for determining recombination rate. Maybe it canalizes or buffers any of the variation that you get at the beginning of the pathway. And it's these genes that regulate this step that are most important in determining divergence and recombination. And we have a little bit of evidence to indicate that maybe these later states are more important. So in mice and in yeast, there's a process called crossover homeostasis that suggests that it is in fact the case that these steps buffer earlier variation and generate and are most important for determining the total number of crossovers that happen in the genome. So I want to just take a step back first and talk about what I mean when I say measuring the rate of evolution. And so one way that you can do this, and the way that's going to be important for the results that I'm going to show you, is we compare the frequency of synonymous and non-synonymous substitutions. So a synonymous substitution is a nucleotide difference between species that doesn't change the resulting amino acid. So here we have a codon that codes for threonine, a specific amino acid. And then say this was in humans, and we looked at the same codon, the homologous codon in macaques, we might find a nucleotide difference. So in macaques, you might see an ACC instead of an ACU. But because our genetic code is redundant, this also codes for a threonine, or the same amino acid. And so we would call this a synonymous substitution. And we make the assumption, which may not be totally valid, but valid enough that we accept the premise enough to use these tests, that synonymous substitutions are not subject to selection. So the idea is that even though there's a nucleotide difference, it can't change the resulting phenotype because it doesn't change the amino acid that's incorporated into that protein. And so selection doesn't really see synonymous substitutions. Alternatively, we might see what we call a non-synonymous substitution when we compare the human genome and the macaque genome at this specific location. And in this case, we may find that in humans, this C is now been substituted as a U somewhere in the evolutionary history between humans and macaques. And this actually does change the resulting amino acid. And so you get an isoleucine instead of a threonine. And we assume that this is much more likely to be subject to selection because this is actually going to change the amino acid and the protein and potentially the phenotype. And so by leveraging the frequency of these two types of substitutions, we can start to make inferences about how selection may be acting on a gene. And so we compare DS, or the frequency of synonymous substitutions, with DN, or the frequency of non-synonymous substitutions. And this ratio can tell us a lot about how we think selection is acting. So if we expect our neutral expectation, or our expectation if selection isn't really doing anything, is that these frequencies of DN and DS should be roughly equal to each other, because that would suggest that selection isn't weeding out or favoring non-synonymous substitutions. They're just like those synonymous substitutions, which we think are invisible to selection. So this is our neutral expectation. Alternatively, what we see most of the time when we looked at genes in the genome is we see evidence that there's a lot more synonymous substitutions than non-synonymous substitutions. And so each one of these dots represents a hypothetical gene that we're looking at. And so each of these cases, the frequency of synonymous substitutions is much higher than non-synonymous substitutions, which indicates that selection is probably weeding out those non-synonymous substitutions, and that any changes to these genes that change the resulting proteins haven't been favored over evolutionary time and have been weeded out. Alternatively, what we see rarely, but it's very exciting, is that sometimes we see more non-synonymous substitutions than synonymous substitutions. And that tells us that selection is acting on those genes to favor changes that change the resulting protein and the resulting phenotype. And so this is what we expect to see in the cases of positive selection or directional selection, where there's some sort of natural selection going on that favors differences in that protein and favors differences between species. Unfortunately, we often don't get such a clear signature of positive selection where this ratio of DNDS is above 1. What we often get are these genes that have high DNDS. We'll call it rapid evolution, but when they're right below 1, it's impossible using this test alone to determine whether that's the result of positive selection or relaxed or neutral evolution, because both of those different evolutionary hypotheses would give the same sort of pattern. So we can use other tests that leverage other types of data to sort of differentiate between those two alternatives when we get into cases like this. So the first project I'm going to tell you about is from the paper that I sent around, or someone nicely sent around for me to the graduate students. And so what we did was we asked whether the rate of evolution of recombination genes, we measured it across the mammalian phylogeny in the context of the pathway. And so we did it by comparing DN and DS of these genes. And we weren't doing a pairwise comparison, we're doing it in the context of a phylogeny. So we're using a program called PAML, which is phylogenetic analysis for maximum likelihood. And those are a lot more complex models of molecular evolution. That the data that they use ultimately is the same DNDS data that I just explained, but these models allow DNDS to vary on different branches of phylogeny or within codons of a gene. And we also then looked at polymorphism and divergence between humans and macaques, because that can help us sort of distinguish between different sources of rapid evolution, different evolutionary forces that can generate similar patterns. And I started this project in collaboration with my postdoc advisor, Dr. Brett Pazer. So the first thing that we need to do was figure out what genes to look at. And so we selected a panel of genes that are involved in each of these different steps of the recombination pathway. So we had a few criteria in selecting these genes. We wanted to select genes that were well described, so we know really clearly what role they have in recombination. We wanted to have somewhat equal sampling across all of these steps, and we wanted to emphasize or make sure that we captured genes which had been associated with within population variation and recombination rate. And then we selected and went into publicly available databases to pull out sequences of each of these genes from a panel of mammals. And so this is a phylogeny, or a representation of the evolutionary history of the mammals that we selected for inclusion in our study. So we have, over here, we have some rodents, some mice and rats. We also included primates. We have a nice panel of sequences from primates and tree shrews, and Laurasithians. So over here, we have a lot of the animals that you associate with agriculture. So sheep, cows, and pigs, and horses, and bats are over here in this part of the phylogeny as well. So we went into public databases, we pulled out the sequences of each of those genes that I showed you in the previous slide, and we aligned them so that we could look at patterns of synonymous and non-synonymous substitutions between species in the context of this phylogeny. We also did, there was an important step in data curation, which is when you do these molecular evolution studies, you have to deal with alternative splicing. And alternative splicing occurs because sometimes the genome, you get the same gene, it can be spliced and processed in a way that you get slightly different copies of the protein. And we wanted to make sure that we were comparing the right copies of the protein. And so we took sequence or expression data sets from testes where recombination occurs and looked at what isoform or splice variant of each of these genes was expressed in the testes and selected that for our downstream analyses. Right, so what did we find? We found a lot of variation in the evolutionary rates of recombination genes. And so I'm showing you here at the bottom, this is an average of the evolutionary rate across that whole phylogeny of mammals. And then up here, it's just a pairwise comparison of humans and macaques. You can see the distribution of DN/DS values, or evolutionary rates, which we can also call omega, are relatively similar, whether we look at just part of that phylogeny, just humans and macaques, or whether we look more generally at mammals. The other thing that you will notice is that these evolutionary rates vary between 0 to close to 1. So we didn't see any genes that had this really strong signature of positive selection such that DN was greater than DS across the whole gene. But we did see a lot, or a fair number, that fell into this part where we would say is indicative of rapid evolution. We also found quite a few genes all the way over here with a DN/DS ratio near 0. And so these genes are ones that have way, way more synonymous substitutions than non-synonymous substitutions. And so they're likely have experienced really strong purifying selection. So this is interesting, but what we wanted to know also is this different than what you would expect if you were to randomly grab genes from the genome. So maybe if you picked any group of genes that you're interested in and you went and grabbed them from the genome, you would see this level of variation and this mean recombination rate, or mean evolutionary rate that's as high as we saw in recombination genes. And we found that this wasn't the case. And so what we did was we took a larger set of evolutionary rates from about 4,000 different genes between humans and macaques, and we randomly grabbed the same number as we did with recombination genes. And we did this about 10,000 times, and we asked how likely, if we were to randomly select genes from the genome, would be to get a group of genes that had the same mean evolutionary rate that we saw in recombination genes. And it turns out we vary pretty infrequent. So less than 1% of the time did we find a group of genes with a mean evolutionary rate that was equal to or higher than what we saw in recombination genes. So that tells us that recombination genes exhibit elevated rates of evolution than other genes in the genome, which is interesting. And then what we also wanted to do was to take this variation in evolutionary rate and map it onto that pathway and ask whether different steps of the pathway or genes that regulate different parts of that recombination pathway evolve faster than other genes. And so this is just another way of representing variation in evolutionary rate and recombination genes. And I've color-coded it here. So genes that are close to the average for recombination genes have these sort of muted reds and blues. And as you get to be, have really low rates of evolution or really strong evidence for purifying selection, they get to be dark blue. And when you have really high rates of evolution or DNDS values that get close to 1, they have these dark reds. And so now I'm going to use this color scheme to map these differences onto recombination rate or recombination pathway. So here's what that looks like. So each one of these cases, I have the gene that regulates that step of the pathway color-coded based on whether it's evolving really, really rapidly in these dark reds or really, really slowly in these dark blues. And if we look at this, we can kind of pick out some patterns. So really highly conserved genes appear to be involved in processing and detecting those double-strand breaks, and that kind of makes sense. And genes that are evolving most rapidly seem to be in those steps of the pathway that are involved, that we think might be likely to generate differences in recombination rate. But we wanted to be a little bit more rigorous in the way that we were connecting these patterns of evolution with different steps of the pathway. And so we wanted to ask whether there were significant differences in the evolutionary rates of genes in each step of the pathway. And as you could kind of guess from looking at this and seeing all of this variation within each step of the pathway, we didn't see significant differences in evolutionary rate when we grouped genes by pathways. So you can kind of see it's a little bit higher. Sorry, I couldn't fit the names of each steps, but this is in the order of the recombination pathway. So these are the genes that are involved in double-strand break formation, double-strand break processing, homology search, and strand invasion, the Synaptonemial Complex, that's proteinaceous structure that stabilizes the homologous chromosomes, the crossover versus non-crossover decision, and the resolution. And you can see some variation between the steps, but it isn't significantly different because there's so many differences between genes in each of those steps. But we also looked at which genes show signatures of positive selection. And so while none of the genes showed a DNDS value that was above 1, these models that we use, PAML, allows you to look for differences in DNDS values within genes. And so it seems very likely that in a big, long gene, only certain parts of that gene are subject to positive selection. And so PAML allows you to ask whether specific codons within that gene have a DNDS ratio, or estimated to have a DNDS ratio, that are higher than 1. And use this as evidence of positive selection. And we saw quite a few genes have evidence of positive selection, or some set of codons within those genes have elevated levels of non-synonymous substitutions. And when we looked at where those genes were located in the pathway, we saw that there was a statistical overrepresentation of genes with signatures of positive selection in these two steps, step 4 and 5. So genes that we sampled that were involved in the stabilization of that homologous pairing and that crossover, non-crossover decision were much more likely to have signatures of positive selection than you would have expected. And so that gives us some idea that it is in fact the case that genes that are regulating this crossover, non-crossover decision exhibit more frequent signature of adaptive evolution and potentially are contributing to differences in recombination rate between species compared to other steps of the pathway. But we also focused in on one gene in particular that really stuck out, which is called TEX11. And TEX11 stuck out to us for a couple of reasons. One, it evolves really rapidly. So it had the highest omega or evolutionary rate in mammals in general, but also in the human macaque comparison. And it had a really robust signature of positive selection. So we went and tried to control for anything that can lead to erroneous inferences of positive selection. No matter what we did, TEX11 always showed us a signature of positive selection, which is quite impressive because a lot of those processes involve throwing away most of your data. And so when you throw away a lot of your data and you still see a significant p-value, it's exciting. And interestingly, we went and learned a little bit more about what TEX11 does, and it directly interacts with two other of our most rapidly evolving genes. It directly binds to SYCP2 which is a protein that forms that proteinaceous complex between the two homologous chromosomes. And then it also directly binds and interacts with this gene called SHOC1, which starts to recruit all of those genes that designate whether a double-strand break is going to be a crossover or not. And so not only does it directly interact, but we were also able to show that the rate of evolutionary rate co-varied significantly between TEX11 and these genes. In other words, when we mapped evolutionary rate onto the phylogeny, we found that on the branches of the phylogeny where TEX11 is evolving most rapidly, SHOC1 and SYCP2 were also evolving most rapidly. And conversely, when TEX11 was evolving slowly, these genes were also evolving slowly, which is consistent with the idea that these are really interacting highly with each other. And also, TEX11 shows strong recombination rate phenotypes in mice and humans. So variation in TEX11 has been associated with variation in recombination rate in humans and in mice. So what we wanted to know next is, can we connect the rate of evolution of TEX11 with variation in recombination rate across the mammalian phylogeny? And in order to do this, we went into the literature and found estimates of recombination rate for as many mammals in our study as we could. And so we couldn't find estimates of recombination rate for all species, so we had to look at a subset of the phylogeny that's represented by the organisms that still have pictures here, where the organisms that we could get measures of recombination rate. And specifically, the types of measures of recombination rate we looked at were from these immunofluorescent cytology studies, which mark specific proteins that designate crossovers and allow you to actually go in and look at these slides and count the number of crossovers per chromosome. And we used a program called CoEvolve to look for correlations between the rate of evolution of TEX11 and the recombination rate across this phylogeny. And to be honest, quite shockingly, given the number of species that we had, we found that for TEX11 and only TEX11, there was a positive correlation between evolutionary rate, or how fast this gene was evolving across the phylogeny, and recombination rate. And so just to point out a few comparisons, since there's a lot going on here. When we look at sister species in the context of this phylogeny that we were using, we can see that this pattern pops out over and over again. So here we have cow and sheep. Sheep have higher recombination rates than cow, and TEX11 evolves a little bit faster. We can also look at mouse and rat, which are sister species in our phylogeny, and we see that pattern again. And here are macaque and humans. So this is a pretty nice correlation, given the data that we had, to indicate that on the lineages where TEX11 is accumulating more changes in its protein, we're also seeing the evolution of higher rates of recombination. So we're able to connect these patterns that we're seeing at a molecular genetic level to these phenotypic patterns that we're seeing across the phylogeny, which is pretty cool. All right, and that's just to come back and remind us about what TEX11 does, which it's this bridge that between this complex that stabilizes homologous chromosomes and the regulatory genes that specify recombination events, which also indicate that it's potentially a really important part of the pathway that can modify or determine how many crossovers occur in a genome. All right. So to summarize, we wanted to know if we observed higher rates of divergence in steps that determine recombination rate. And we found that we had a stronger signature, statistically significant signature of more adaptive evolution of regulatory steps that regulate these early steps of the crossover, non-crossover decision. And specifically, we found some really interesting results about how TEX11, the molecular changes in that gene, could be associated with phenotypic changes in recombination rate across the mammalian phylogeny. So now I wanted to switch gears a little bit and talk to you guys about the cool projects that my lab is working on right now. So one area of research that, to be honest, I wasn't necessarily planning to do when I started my position here, but I got really inspired to work on by talking to some of my new colleagues, specifically in collaboration with Dr. Federico Hoffman, who does some really cool work on gene family evolution, and specifically has some really cool stuff that he's done on hemoglobin. And Dr. Jean-Francois Gu, who has a lot of experience working on the consequences of whole genome duplications. And one of the questions that we wanted to look at is, what are the consequences of whole genome duplications on the recombination pathway? So why would we expect there to be some important interaction between whole genome duplications and recombination? So whole genome duplications are mutations that result in the duplication of the entire genome. And a consequence of that is that directly following whole genome duplications, instead of having 2 sets of homologous chromosomes, now you have 4 sets of nearly homologous chromosomes. And when we think about that cell and how it lines up and pairs these homologous chromosomes, it's going to immediately run into trouble, because it's not going to be clear during homology search and recombination whether this chromosome should be paired with this chromosome or this chromosome or this chromosome. And what happens is that you get, whenever you have more than 1 crossover per chromosome, you run the risk of getting these tangles, where this chromosome has a crossover with this chromosome, and maybe there's another crossover going on with this one down here. And as you can imagine, when the cells starts to pull those chromosomes with the spindles to get equally represented in the 2 daughter cells, it results in a big mess. And so there's really integral relationship between what happens post whole genome duplication and the meiotic recombination pathway. And one way in which the meiotic recombination pathway might respond to these challenges is to reduce recombination rate, because the fewer times you have these crossovers, the fewer chances there are for these tangles to form. And one way to reduce crossover frequency is to reduce copy number of these genes. So all these recombination genes in the pathway itself are also duplicated. And so we hypothesize that you should see a rapid loss of paralogs or duplicates of recombination genes following whole genome duplications. So we wanted to know whether these paralogs, or these new copies of recombination genes, are lost at higher rates than expected in the genome following whole genome duplications. And we can use vertebrates as a system to look at this, because vertebrates have experienced multiple rounds of whole genome duplication in their evolutionary history. So back in the early evolution of vertebrates experienced 2 rounds of whole genome duplications. And then there was another round of whole genome duplications in teleosed fish. And this allows us a natural replicate to look at 2 instances of whole genome duplications in vertebrates and ask whether, after following these whole genome duplications, we see this rapid loss of paralogs of recombination genes. And we can use phylogenies to reveal the duplicative history of gene families. And so here you see a phylogeny. We have a bunch of different organisms, and each of these colored bars represents a whole genome duplication event. And so if every single paralog of a gene that occurs in response to a whole genome duplication is retained, we should, for this hypothetical gene, see lots of copies of that gene, lots of paralogs in that gene that replicate that duplicative events, or those consequences of whole genome duplications. So if we start here and we look at this teleosed gene, we should see a paralog. Paralog is, anyway, you know what I'm saying. That was a tongue twister. Another copy of that gene that originated through the whole genome duplication in teleosed fish. And if we go farther back, we should see the retention of paralogs from the previous whole genome duplication, and so forth. So it leaves a pattern in the phylogenetic relationship of these genes. And if these paralogs are rapidly lost, what we should instead see are these one-to-one relationships. So every time these genes were duplicated through a whole genome duplication event, they were lost. And so the resulting tree is much, much simpler, and it replicates the species tree. Alternatively, we can also have cases of hidden paralogs, in which case the duplication event, the duplication of the gene was retained longer in evolutionary history before being lost. And so in this case, the natsistome gene copy here was from a different paralog than the teleos genes that were retained in this hypothetical gene. And if we compare these two, they can tell us some relative differences in the timing of the loss of these paralogous genes. So if it happens really quickly, we shouldn't see any hidden paralogs, whereas if these duplicates are retained longer in evolutionary history, we can see these more complicated patterns of hidden paralogs. So we begin to look at this, and we've seen that there's very little evidence of paralog retention among recombination genes. And so this is not a recombination gene that I'm showing you first, but it is a gene that's involved in cell cycle regulation. So it has a similar function as recombination genes. And this is SPO11. So SPO11 is a recombination gene that initiates those double strand breaks. And what we can see by looking at the phylogeny of SPO11, which we've reconstructed, is that every time there's been a duplication event, that duplicate gene has been lost, indicating that it's consistent with our hypothesis that paralogs of recombination genes are lost more rapidly following whole genome duplications. And we are currently working on expanding the number of genes that we're looking at and being a little bit more rigorous in how we compare the rates of retention and recombination genes to control sets of genes. So that's an exciting project that we're working on right now. And I also wanted to highlight another project that my graduate student, Austin Drury, is working on, which I think he's getting some pretty cool results, which is modeling recombination rate as a quantitative trait. And the idea behind his project is that if we can simulate the evolution of recombination rate with realistic genetic architectures, like multiple genes and a strong component of environmental variation, we can apply these models to data from real populations and learn about these different hypotheses or different ways in which evolution may be acting on recombination rate. And so Austin's built this model. He's done a great job using some pretty awesome computer programming skills to modify a program to simulate the evolution of recombination rate. And he's applying that model to human data. So there's this great data set on within population variation in humans from Iceland. And here you can see a distribution of female recombination rates from that study. And the first hypothesis that we've looked at is whether that lower bound of recombination, which occurs because if you get any less recombination than that, you don't get chromosomes accurately segregating into each daughter cell, whether the distribution that we see is consistent with that lower bound, or whether there's evidence that selection is still acting on recombination rate in humans. And so when he's run these simulations, he started the population off where it has a trait distribution that's similar to humans, and then exerted selection in his model of that lower bound and looked at how that trait distribution evolved. And what he's seeing here, so here you have in blue is the human, female human data, and here in orange is his simulated data. And over time, through his simulation, you see the distribution in the simulated data move farther away from that lower bound, which indicates that selection is actually still currently exerting selection on recombination rate. And so we should expect one of two things. One possibility is that recombination rate as humans is still evolving and under selection, specifically to be a little higher in human females. The other possibility is that there is some upper bound or some selection that's keeping recombination rate from evolving and responding to selection. And so he's building some more complicated models with different fitness landscapes to sort of differentiate between those two alternatives and to understand what an upper bound to recombination might look like. And so with that, I want to acknowledge my collaborators that I highlighted throughout my talk and where I've gotten funding from, and see if you guys have any questions.
Anyone have any questions?
Yeah, I guess I have one. Again, thank you for accepting student invitation and for coming today. Just have a quick question. Do you think that the divergence you are seeing in the recombination genes seems to be advantageous? Like for the positive selection of TX11, is it beneficial to the evolution of those organisms?
Yeah, so the answer is I can't tell for sure from my molecular evolution studies. And it certainly seems to be driving some of their variation in recombination rate based on the correlation between rate of evolution and recombination rate, but it falls in this really difficult area where it could be due to relaxed selection or it could be due to positive selection. When we use the more rigorous McDonald-Kreitman test to leverage polymorphism and divergence, the data there, again, was very hinted at a role for adaptive evolution or natural selection, but it wasn't quite strong enough that I could say with total confidence that the signature is due to adaptive evolution and not neutral forces.
Okay, thank you.
I have some questions if there's time, and no one else has any. So the first, as you were going through the talk, I was thinking about this, and I know that biological systems tend to be very redundant. And I was thinking about your pathway analysis, and I was wondering if people have thought about this or if there's a way to model multiple genes or even multiple regions within genes together to see how the balance between purifying and possibly adaptive selection might be working in that network to kind of shift the bounds to the upper or lower limit. Because it's really interesting about TX11, but it's a little surprising that only a single gene could, shift the whole net result of the pathway.
Yeah, so I think one reason that it's hard for us to give a really definitive answer on whether adaptive evolution is driving this is because signatures of adaptive evolution are really hard to pick out in a statistically significant manner unless they're exceedingly strong. And what I could tell from what I've done, which I think sort of hits on your question, is we can look at whether the rates of evolution of different genes are correlated with each other. And that tells us whether there's some evidence of coevolution between genes. And so we did find a few patterns that are suggestive that this is it's not just TX11, but it has to do with the context that it's evolving in the pathway. The first is that if you look at recombination genes in general, their rates of evolution are more correlated with each other than you would expect in the genome. So there's an elevation in correlation of evolutionary rates among recombination genes. And in particular, if you look at just that triad of those three rapidly evolving genes that directly interact with each other, they show statistically elevated rates of covariation, or evolutionary rates are more correlated with each other than among the recombination genes. So it definitely seems to be that there are coevolution between TX11 and those genes that it's directly interacting with. And beyond that, there's some evidence of positive selection acting on a bunch of different genes. So it's definitely not one gene alone that's driving this. And it does seem to be the case that genes involved in specific steps of the pathway are more likely to be driving, are more correlated with each other and more correlated with the variation that we see in recombination. Does that sort of get at what you're asking?
Yeah, it's just, I guess it becomes a complicated question too, to try and put multiple bits and pieces into the model. But yeah, it's really interesting. If no one else has a question, I have another question. I was really interested in that last part of your talk. You were talking about the whole genome duplications. And I've been working with some collaborators who have been interested in leveraging the long reads and the linked reads to try and get a better understanding of structural variants, which surprisingly are still very hard to quantify. And I was wondering if you could speak to how not whole genome duplication, but structural variants might contribute to recombination and possible evolution of recombination rates.
Absolutely. There's a lot of things that I could go on from that question. One thing that I just want to point out to the audience in general, which I think is really important, which I didn't talk about, is that one really quick way to reduce recombination rate is to get an inversion. So if you have a mutation that creates a genetic inversion, then those parts of the chromosome can't pair properly, and that reduces recombination. And that's also a really important step in potentially in evolution of sex chromosomes, but also it's been seen in areas of the genome that are responding to local selection adaptively. You can see that they're sometimes in areas of inversions, which prevents recombination from mixing up co-adapted allele complexes. So that is super important. Another aspect that I kind of